And I'm actually going to start by thanking my group. And one person I'd really like to thank is Yuja King, who's in the audience, and she's led the two programs that I'm going to talk about. And here we all are in, not actually in Oxford, but as you'll see, the Oxford group is very China-friendly if you look at the photographs carefully. Okay, so nanopore sequencing was developed initially in academia, a lot of the work of getting the DNA into the pores, finding enzymes that would transport single strands through the pore, identifying the bases and so on, were done in a quite primitive way in academic labs. And then Oxford Nanopore, that was founded in 2005, finally went on to perfect the sequencing, which was a huge amount of work, and make it highly parallel. So around 2012, we ended up with the first version of the MinION. This is a very inexpensive device. It's portable. It's very user-friendly. And you can do ultra-long reads, megabase reads. And the initial system was parallel up to the extent of about 500 pores. And nowadays, the accuracy is pretty good. But what we'd really like, in addition or as a supplement to the MinION, is something like the tricorder that's you've seen in Star Trek. So this device will tell you everything that's wrong with the patient in about 5 seconds. So that's where we'd like to head eventually. And genome sequencing is all very well, but it really doesn't tell us everything about a cell or a tissue. And with that in mind, we've been looking back at small molecules, which is how we really started with nanopores. And more recently, we've also been looking at polypeptides. So back when we started working with protein nanopores, we developed a technology called stochastic sensing, which was based on the single molecule detection of small molecules by non-covalent interactions. So we would measure the ionic current that flowed through a pore, and then with a molecule bound, that current would change, and this would be a reversible process, so we see binding coming off, binding coming off, binding coming off, and so on. And the frequency at which these events occur tell us the concentration of the analyte, and then the actual shape of the events themselves, the amplitude, the lifetime, whether there's noise during the event, and so on, helps us to identify the analyte. And of course, that became the basis of identifying the GAT and C bases in DNA. But initially, when we founded Oxford Nanopore in 2005, it was to look at small molecules. And I'll just give you one early example of that. Here we took the hemolysin pore, engineered it, it's a 7 subunit protein, to make 2 rings of arginine inside the pore. It's a very primitive engineering because the signal actually tells us the identity of the analyte. We don't have to make a very, very selective binding site. And we use this to monitor IP3 in cellular fluids. So the idea is this is a single pore here, 2 different states. It either combines something that interferes with what we're doing, in this case ATP, or it combines your analyte. And because this is a single molecule technique, these events are separated in time, we don't have the problems that we would have with an ensemble method. So as you can see here, here's the current flowing through the pore. We throw in ATP at millimolar concentrations. ATP would interfere with our measurements. And then when we add a tiny amount of IP3, we can see these distinctive binding events that are longer and have a larger amplitude, and you add more, you see more of these events, and so on. So this is a very, potentially a very powerful method. So in terms of building the tricorder to look at what's wrong with people, there's been a lot of interest in sensing volatile organic compounds that might be different in people who are sick. And the idea would be that you would take these volatiles, get them into an aqueous solution, and then identify them as they went through the nanopore. Now, the problem is that healthy humans will release about 3,000 different volatile organics, and we had to reduce the subset of molecules that we're looking at. And one way in which we've been doing that is by using reactive analytes. So, let's go back there. Let's get the right button. So to reduce the recognition problem, we look at a subset of volatiles, which are aldehydes. Humans release only about 200 aldehydes, about 7% of the VOCs, and we're detecting these by reversible covalent chemistry. So let's say you've got a lot of molecules that someone's breathing out. We just want to look at a subset of them. And the way we're doing this is building a pore that has a thiol group inside it. So it just has a single cysteine in this heptameric pore. And aldehydes will react with the cysteine reversibly to form a hemithioacetal, and chemistry is shown here. And so the idea is if someone is breathing out one of these compounds, and this gives a unique amplitude, you'll see a series of events that will help you identify this substance. Now, this is even more interesting because the molecules, when the aldehydes react, they form two different diastereoisomers, and they have actually different extents of block of the current, much more subtle than is shown here. But this is very important because you can only distinguish so many different current levels, so you want more information to allow you to identify the compounds that you're looking at. So we've made use of this as well. So just, again, a little bit of detail. To get this to work, we have to engineer the pore extensively to remove residues that might also react with aldehydes. We put in a single cysteine. It will react with that aldehyde and form this adduct reversibly. This will be coming on and off the protein. And this works amazingly well. So in this example here, we're just looking at a series of aliphatic protons, aliphatic aldehydes, and they can all be distinguished from each other by the amplitude of the current. And if you look very carefully, you can actually see the diastereomers. So in this case of heptanal here, there are two distinct current levels for the adduct. So that allows us to look at these molecules in really quite complex mixtures. So that's one example I wanted to show you. The second one involves looking at proteins rather than nucleic acids. So again, as you will know, there are only about 20,000 protein encoding human genes. They make a large number of transcripts. We're really not sure how many, but we know that there are millions of different proteoforms, either formed by post-translational modification, alternative splicing of RNA, and so on and so on. So what we'd like to do using nanopores is to count the various protein species, map post-translational modifications. I don't think there's much purpose in actually sequencing proteins in this way. We, in general, know the sequence from the genome. And this is a single molecule technique, so we'd like to bring it down to the single cell level eventually. So the first problem is, how do you get a protein to go through the pore? Initially, when we started nanopore sequencing, we're using DNA. DNA is negatively charged. We would just apply electrical potential and pull it through the pore. Of course, you can't do that with a protein. Again, the idea is to post-translation modification, pull the protein through the pore, and identifies it as it goes through. And what we've been using is a process called electroosmosis. So we make a pore that's ion selective, so it will just, if you apply a potential, only one type of ion will move through the pore, so this is directional. The ions carry the solvent, water, with them, and this produces a flow that's surprisingly strong, and the force is up to, say, 10 piconewtons or so, which is just what you need to unravel a protein if you pull at the terminus. So I'm just going to show you some really initial experiments. What we've done is make concatemers of thioredoxin, a protein that we'd worked with before, and these concatemers are over 1,000 amino acids in length, so we're not just looking at small peptides now. And again, the idea is that if there's a modification here, we will identify it as it goes through. So if we take one of these concatemers that has 7 subunits in it, we will actually see this 8 subunits. This will enter the pore, and you'll see an event each time the thioredoxin goes by. And this was actually the initial proof that we could get a very long polypeptide chain to go through the protein. And each of these events has a distinctive set of current levels, and the way we interpret this is that initially the linker between the thioredoxins is in the pore, then the protein partly unfolds, and this is level 2, and level 3 is when the protein completely unfolds. At this point, the electroosmotic flow can't really quite keep up with the uncoiling of the protein, and we get a complete block of our nanopore. So what we did in the lab was to put a whole bunch of different post-translational modifications into this model protein, phosphorylation site, glutathionylation site, and small oligosaccharides. And these would produce very distinctive signals. So when glutathione is attached, for example, instead of the current being at this level here, you get a very distinctive current level. And won't go through all the different experiments we did, but we can distinguish all these different modifications at different sites within the protein. And it's important to note that, say, a phosphorylation site can be identified deep within a polypeptide chain. Again, we're not looking at short peptides here. And phosphorylation sites that are really quite far apart, in this case almost 300 amino acids apart, can be identified. Now, as you're probably aware, you're thinking, gosh, there are hundreds of different post-translational modifications. How are these people going to deal with that? Well, one thing we're looking at very intensively is to look at binders for different post-translational modifications. They could be nanobodies, for example. But the idea is that you'll have these specific binders for specific modifications. If the binder is large, it will hold up the polypeptide at the mouth of the nanopore and eventually get pulled off. If they're very small, they will get translocated through the nanopore and produce a distinctive current level. And we're making a lot of progress in that area at the moment. So basically, I'm just trying to tell you there's a lot more than you can do with nanopores than sequence DNA and RNA. You can look at small molecules, you can look at polypeptides, you can look at oligosaccharides, really anything that dissolves in water, and as biologists we know that all important molecules dissolve in water. So, so really now we can say we can look at nucleic acids, polypeptides, small molecules, many other things, and we're maybe on the way to making something like this tricorder. I would say if you look at it compared to the MinION, it's rather bulky and has a lot of different buttons that would confuse someone like me. So I think in the end we'll make something you can put in your pocket and is much more user-friendly. So let me just finish here by thanking my group again, and thank you for listening.